Namaskar. Today we will study apoptosis. Apoptosis is programmed cell death, where the unwanted cells are eliminated from our body. In apoptosis, the first thing which occurs is the cell shrinks. The cell shrinks and there is formation of the apoptotic bodies. The apoptotic bodies, and these apoptotic bodies on their surface they will show phosphatidylserine. Phosphatidylserine, they will express on their surface phosphatidylserine, which will be recruiting the macrophages, and these apoptotic bodies are now phagocytosed by the macrophages, and there will be no inflammation. There is no inflammation. So thing is, cell shrinkage occurs, formation of blebs occur, cell shrinkage, the formation of bleb-like structures, blebs, and there is nuclear fragmentation, and these blebs on their surface will express phosphatidylserine, which are called as apoptotic bodies, and these apoptotic bodies are then phagocytosed and there is no inflammation. And the mechanism of apoptosis is there are two pathways. One is the classical pathway and the other is the non-classical pathway. The classical pathway or classical caspase-dependent apoptosis and the caspase-independent apoptosis pathway. The classical caspase-dependent apoptosis pathway is of two types, the extrinsic pathway and the intrinsic pathway. The extrinsic pathway, there is engagement of transmembrane receptors by its ligands. The external factors, these ligands will induce this pathway, and the intrinsic pathway is the mitochondrial pathway. The mitochondria involvement is there, and the caspase independent pathway is also there. So let us first discuss the extrinsic pathway, where the ligands can be the FAS or APO1, tumor necrosis factor alpha, and TNF related apoptosis inducing ligand. So accordingly, these ligands have receptors on the cells called as the death receptors. So APO1 or FAS will have CD95 or APO1 or FAS receptor. TNF alpha is having TNF alpha receptor. TRAIL is having TRAIL receptor. And what happens, the ligand like FAS or TNF alpha can bind to their respective receptors. The death receptors, say FAS binds to FAS receptor, TNF alpha binds to the TNF alpha receptor, and similarly the TRAIL binds to the TRAIL receptor, and there will be oligomerization of these receptors. Mainly the trimerization will be there, and they will recruit the adapter proteins. So in case of FAS, there will be FAS associated death domain, FAD, this is the adapter protein here. In case of TNF alpha receptor, there is TRAD, TNF receptor associated death domain. So these are the adapter proteins which will be recruited, and these adapter proteins will induce the death inducing signaling complexes, which will activate the pro-caspase-8 to caspase-8. So the caspase-8 is the active form of the pro-caspase-8, and caspase-8 then activates pro-caspase-3 to caspase-3. This caspase-3 causes the degradation of cellular and nuclear proteins, and there will be breakdown of the chromosomal DNA, chromosomal fragmentation will be there, and that will cause apoptosis. So the nuclear fragmentation will be there, and these apoptotic bodies will phagocytose, and then there will be apoptosis. The intrinsic pathway is regulated by three classes of BCL2 family of proteins. One is anti-apoptotic family and two are the pro-apoptotic family. Anti-apoptotic BCL2 family of protein is BCL2, BCL-XL. Pro-apoptotic effector BCL2 family of protein is BAC, and pro-apoptotic BH3 only proteins are BAD, BEAM, BEAD, PUMA, NOXA. These are the pro-apoptotic factors. So pro-apoptotic means apoptosis inducing family of proteins. One is from BCL2 family BAC, and other is from BH3 family BAD, BEAM, BID, PUMA, NOXA, etc. And anti-apoptotic BCL2 family protein is BCL2 and BCL-XL. Then in the intrinsic pathway is stimulated by the apoptotic stimuli, like lack of survival stimuli, when there will be withdrawal of the growth factors, when there is hypoxia, when there is presence of genotoxic substances. P53, the negative regulator of the cell cycle, all of them act as apoptotic stimuli, and they activate BACs, the apoptosis, pro-apoptotic effector BCL2 family protein BACs. And BACs enters into the mitochondria, and in the inner membrane space there is presence of cytochrome C, and BACs causes formation of oligomeric pores through which the cytochrome C will be released, or leakage of cytochrome C will be there. And this cytochrome C, in presence of ATP and apoptotic protease activating factor APAF1, forms the APAF1 cytochrome C complex, and this complex, in presence of caspase recruitment domain CARD, forms a assembly, and that assembly is called apoptosome, assembly of apoptosome. And this apoptosome also contains the pro-caspase-9, which is converted into caspase-9. So in the extrinsic pathway we have caspase, we have caspase-8, whereas in the intrinsic pathway we have caspase-9, and this caspase-9 converts pro-caspase-3 to caspase-3. So ultimately caspase-3 is activated, which causes the degradation of cellular and nuclear proteins, and there will be formation of blebbs, the nuclear fragmentation, breakdown of chromosomal DNA, and that will result in apoptosis. So these are the caspase-dependent pathway. So the crosstalk between the extrinsic and intrinsic pathway is, there is the third group of that, the second group of pro-apoptotic BH3 only protein BID. This BID is converted to TBID by caspase-8. So in the extrinsic pathway there is production of caspase-8, which activates the BID to TBID, and this TBID, like BACs, can enter into the mitochondria and cause release of cytochrome C, which will form the apoptosome, and there will be formation of caspase-9, and ultimately there will be formation of caspase-3. Caspase-8 can convert pro-caspase-3 to caspase-3, caspase-9 can also convert pro-caspase-3 to caspase-3, and the link between, the crosstalk between the extrinsic and intrinsic pathway is caspase-8 of extrinsic pathway can activate BID and convert BID to TBID. TBID can release the cytochrome C and can activate the intrinsic pathway. So this is the crosstalk between the extrinsic and intrinsic pathway. Now let us see the caspase-independent pathway. And in the caspase-independent pathway, again there is the role of this cytochrome C. So either BACs or TBID, they release the cytochrome C, and release of cytochrome C also induces release of apoptosis inducing factor, a flavoprotein AIF, and endonuclease G protein from this mitochondria. So now we have cytochrome C, AIF, and endonuclease G, which all of them, they will migrate to the nucleus, cause the chromatin condensation, and there will be apoptosis. So this is all about the mechanism of apoptosis. Thank you.